All right, it is 6 o'clock, so I think we'll go ahead and get started. Welcome everyone to the Thrive series. My name is Amna Hussein. I am the Community Outreach and Engagement Manager at the Masonic Cancer Center at the University of Minnesota. On behalf of the Thrive Planning Committee, I'm happy to welcome you to the Fall 2023 series. If you have attended a Thrive program before, welcome back. If you are new to the series, we are so happy to have you here. We are delighted to have such great sessions leading up to tonight and look forward to spending just 2 more Tuesdays with you. Before we get too far, I want to remind everyone that today's election day. So if you haven't already, we encourage you to go and vote. Here in Minnesota, polls are open until 8 p.m. and we encourage you to vote and instead join us live later after you vote, or you can watch the recording of tonight's session and we will post the recording on our website at c.umn.edu/Thrive series. The Thrive program is a collaboration between mHealth Fairview and the Masonic Cancer Center at the University of Minnesota. We are proud to offer 6 sessions this fall. Today is the 5th session. So don't forget to mark your calendar for our final session next week on November 14th. You can register for the last session or rewatch the previous sessions on our website. Again, that's c.umn.edu/Thrive series. Tonight, we will be hearing from Dr. Veronika Bachanova. Dr. Bachanova is a professor of medicine at the University of Minnesota, and her clinical practice centers around patients with blood cancers such as lymphoma and leukemia. Her research focuses on the design and development of new approaches which utilize the immune system to attack cancer cells. She's here tonight to share her expertise with us. Before we get started, I want to just take a moment to locate, or for you all to get familiar with locating the mute and the chat buttons. During tonight's presentation, please use the chat box as well as the Q&A feature to submit your comments and questions. At the end of Dr. Bachanova's presentation, we will have plenty of time for Q&A, and at that time, if you wish, you can unmute yourself to ask your question. If you are a University of Minnesota employee or spouse participating in the university's employee well-being program, we're going to put a link in the chat shortly for you to complete a survey to mark your attendance today to earn your well-being points. All right, without further ado, I will now pass it over to Dr. Bachanova. Dr. Bachanova, please take it away.
Thank you so much. Good evening, everyone. I'm really thrilled to be here. I was invited by the team to talk to you about a very different topic. I realized that this is a new topic and it's maybe very heavy, but I'll try to really introduce this to you in a way that will relate the novelty and in some ways the revolutionary aspect of the cell therapy. So I will share my slides. If people are going to be muted during that, but please use the chat and questions and answers to interrupt or to really try to get the clarifications from me. I am hoping this is more interactive, but you know, if the people want to talk after the session, there is an opportunity to unmute and we can have more discussion. So I will start with sharing my screen. So my task today is talk to you about, sorry about this, let me see, we need to go on a first slide, about cellular therapy. So what is cellular therapy? Is treatment which is using cells. And cells are capable to provide anti-cancer activity. And this is which I will explain to you the aspects of this. So I'd like to describe a revolutionary novelty of using immune cells for cancer therapy. I'd like to teach you about a first genetic cell therapy, which is ever being approved for human disease, called CAR-T. In my presentation, I will share with you that this therapy is curative, which means that it can provide the cure for some cancers, and it serves as an example where the immunotherapy and cell therapy can go. We will learn about very unique adverse effects associated with this therapy. And then I will share with you some challenges this field faces. So what do we hope as oncology practice and oncologists to achieve from our therapies? We want our treatments to be effective. We should have a promise of cure with therapies which we are developing. Ideally, we want this therapy to be safe. We are hoping it is well tolerated and it can be given in any age group, patients, older patients, as well as young and children. We hope that the treatments would be without long-term consequences. And they have to be convenient, or at least it's one of the bars to reach that the therapy should be convenient to administer and then more accessible to all patients who need them. So this is sort of a very high bar, and we are not there, but these are the things we in oncology think about as we are developing new therapies. What are the treatments available for heme malignancies? So we have a chemotherapy, as many of you have experienced, or maybe you had received chemotherapy. It's the oldest and most traditional way to treat cancer. There is a radiation therapy, which is also very old, probably older than chemotherapy, and it's very effective for some treatment, for some cancers. The more novel therapies is immunotherapy, so using immune system and various immunotherapeutic reagents. That there are something called targeted therapies, which are more synthetic therapies developed rationally to treat cancer. There is a treatment called stem cell transplantation, either autologous, which is using your own stem cells, or allogenic, using somebody else's stem cells. And then a very new treatment option, which is really available only for the past 5 years, is cellular therapy. So how can we get rid of cancer cells? We can get rid of them by poisoning them. And that's the chemotherapy and inhibitors. These are cellular poisons. But there is this new paradigm in oncology that, in fact, we can eliminate cancer cell by engaging immune system. In fact, I'll explain to you that immune system role, ever since we are born, is to prevent us and protect us from cancer. The arms of the immune system include antibodies, immune cells, and various immune proteins. So as you can imagine, this is what cancer cell could look like. It's a round structure. Lots of cells make tumors. And these cells have various small proteins on the surface. And these proteins can become what we call a target. Each of them can become a target of therapy. And one of the most traditional immunotherapies is a drug called rituximab. And some of you, if you had a lymphoma or some hematological malignancy, you could have treated with the rituximab, which is the most historical, old-fashioned immunotherapy ever developed. It's an antibody against one of these targets, particularly this one, CD20. So there's a concept of immunosurveillance. It's a long word, but what it really means that immune system surveils against cancer development and protects us. Immune system has two main functions. It fights off infection, but also it search and destroy abnormal cells. And these are particularly cancer cells. They do change somehow in this process of a cancer transformation, and immune cells can recognize this transformation and sense immune cells such as T cells or another cell called natural killer cells. The immune system wakes these cellular fighters and sends it to cancers to fight. There are also antibodies, which are produced by our body to fight off infection, but also they can attack cancers. So in our body, these two arms of the immune system are quite effective in preventing us from developing cancers. However, with the mechanisms which are not entirely clear or understood, at some level and in some people, the immune system fails and it becomes weak and unable to provide this basic function. And a failure of immune system then later, over time, leads to development of cancer. So immunotherapy is really this new concept, new idea that perhaps there are ways we can unleash the weakened or strengthen the immune system to eliminate the cancer. So that's the goal. How do we do that? We can improve the potency or the strength of patient's own immune system. And that's several therapies are able to do it. We can infuse immune systems from a healthy people, from a third party, healthy donors, or young people who have a healthy immune system. And that's one of the therapies which we are using is the infusion of the immune cells, which are strong. We can replace the entire immune system of a patient. This is a rationale of doing a stem cell transplantation. And then we can deliver certain engagers. It's I think, a very well developed term because it really engages patient's immune system to kill cancer cells and attack them. It's engaging them. So these are the 4 different concepts which are being developed all the way from the bench in the basic research laboratories. And over the last decade, most of them have been brought to the clinical trials. And some of them have been proven to be so powerful and helpful that in fact they become approved by the FDA and became a standard of care treatments for some malignancies. So here are some therapies which can use immune system, and these are just examples of how you can modify the antibodies and different immune components to become more powerful. So if you look at this cell, it's a tumor cell, and you can attack it from the surface by different ways. You can use antibodies, you can attach a radiation to the antibody, you can attach a poison to antibody, you can use a effector cell with some engager. I mean, this is how complex this field has become. What I want to talk to you mostly about something called CAR T cell. And I think that it's become such a, revolutionary term that this has become a topic for the general public to learn about because it really changed the face of oncology. And I think to learn about it, why is it important to learn about it? I think it's because it really can become available for most of the cancers in the future. And we are just starting to use it for hematological malignancies. So what is the CAR T? If you look at your normal cell, one of the cells in your blood called T cell, that cell can be genetically modified by sticking a piece of DNA into the nucleus of this cell. And this DNA is called CAR. It's a chimeric antigen receptor. And with this modification, you have a new cell called CAR T cell. This process is called genetic modification. Just like a food can be genetically modified, we are not talking about the genetic modification of the immune system to become a cancer tool, anti-cancer tool. So we also call CAR T living drugs. And the reason for this is that, as you can imagine, the treatments with chemotherapy or some pills, you have to take this every day in order to work. But the cells are alive. They can make offsprings, they can make daughter cells, and each cell looks the same as the mother cell. They can persist in the body for a long time. They need nutrients, just like any other cell, your food, but then they can sustain their life. And so CAR T cells can stay in the body after a single infusion for many weeks, potentially months, and actually even for years. The CAR T cells are also serial killers. What does that mean? That means that once they see the cancer cell, they provide the signals to kill it. But once that happens, they move to a next tumor cell and next tumor cell. And they can continue to do this until the tumor is completely eliminated. And here is Here's the demonstration of how a single infusion of the CAR T cell could eliminate very extensive cancer. The black areas in this PET scan, it's a body PET scan, which we use for lymphoma. The black areas are the lymph nodes. Each of these dots is the lymph node. And as you see, some of them are so large that they make these large tumors under your axillas, in the belly of the patient, in the groin of a patient. Unfortunately, this patient has had many therapies, including chemotherapy and a bone marrow transplant, and his disease kept coming back. After a single infusion of the genetically modified CAR T cell therapy, the patient achieved a complete response. Every single area have responded to treatment. This is the heart, so it's not really a cancer, and this is a bladder, it's not a cancer, but all these other areas, as you see, are really cleared up. So this is how remarkable this therapy can be, and why it brought such a revolution in our understanding of the powerful potency of immune system. So currently, the CAR T cell therapy is approved for several diseases. The main disease is called non-Hodgkin lymphoma, and there are many types of non-Hodgkin lymphoma. But as you see, this is one type, large cell lymphoma. This is a second type, follicular lymphoma, and this green is 3 types. So 1, 2, 3, almost 75% of non-Hodgkin lymphoma histologies are now approved for CAR T cell therapy. So it really changed significantly the outcome of patients with this difficult disease. With this slide, I wanted to impress on you that the progress in oncology takes a long time. And it's because the research is a complicated process. And there are discoveries which are important, which are taking place in the laboratories, typically in academic institutions. And here is, for example, a development or discovery of a target in 1988. This was the very first thing which was necessary for anything else to happen. And then the first CAR Ts were generated in 1993 in a laboratory. But the laboratory discoveries are critical, but as you know, it takes a long time to bring the lab discoveries to the patient bedside. So for this particular agent, it took from 93 to 2017. And this is one of the reasons why we encourage and we are so cognizant of the fact that it takes a team to develop drugs. It takes scientists, it takes investors, but it takes patients, brave patients with courage to enroll into the trials and to understand that without their contribution and their team playing in this process, the things are just not happening. And so, although we have a very effective therapies now, we still are not where we need to be. And even at the University of Minnesota, we have a number of clinical trials to improve on the therapies which are currently approved. So with this, we currently, since 2017, have a much more rapid approval, or now 6 different CAR T products which have a complicated names, and the pharma or industry names are down here. And there are 2 main indications. One is the non-Hodgkin lymphoma and acute lymphoblastic leukemia, and then multiple myeloma for these 2 other products. And I'm not going to go too much detail with this. I just want to impress of you that it's quite quite a lot being done in terms of bringing these therapies to patients, and we have a lot of indications now, including the multiple myeloma, pediatric B-cell acute lymphoblastic leukemia, adult, and then many of the products are now approved in these indications and approved by the insurance, including the Medicare, for patients who are at the line of therapy which is consistent of the, which can bring benefit. So I'm going to skip this because this is just kind of showing some biology. I think you'll be interested to know that the way how these new drugs are delivered to the cells are through the viruses. So not all the viruses are bad. We need viruses to deliver the DNA into the cells, but these are inactive viruses, and they are all helping us to genetically modified cells. There is a big interest to understand the long-term effects of the genetic modification of the human cells. For at this time, there is not known long-term the arrangement of the genetic of the DNA of a patient or any kind of a long-term effects, but it's certainly something very important. And every patient who is treated with a CAR T-cell therapy is going to be monitored for 15 years for development of a secondary cancers or any other long-term complications. So only several centers in the USA are approved to treat with the CAR T cells because the treatment is complicated and require quite an expertise. At Minnesota, there are 2 centers. It's us at the University of Minnesota, and it's a Mayo Clinic. And the centers have to go through a certification process, which is quite lengthy, and are annually audited by the several accreditation organizations to make sure that they are well certified and their staff is all trained in administration of the cell therapy. So how does the process work? Your doctors have to refer you to the cell therapy center or to BMT. We are very much working with the referring oncologist and are able to see patients within a week. The patients have a consultation with an expert in the field. We work with the insurance to get a prior authorization. And then there is a certain process of getting the patient through the therapy. The first step is to make sure the patients are healthy, they have a good organ function, and then they go through the process called apheresis. This is the machine which is removing the T cells from the body. It's a very simple procedure, takes only about 4 hours, and it's quite, quite well tolerated. And the result of that is that we have a T cells selected. The T cells are then sent into manufacturing facility. So it's outside of the body, the cells are manufactured, and they are treated with some cytokines, and basically CAR is a product of its manufacturing. This takes a while. It takes anywhere from 14 to 21 days to manufacture cells. And during that time, the patients have to wait. And then we get the product in a bag. It looks like a transfusion. So during the manufacturing, this is how the CAR-T product looks like. This is that living drug. This is the cellular therapy. It's an IV product. So during this manufacturing, we have to take care of our patients. And how we do it? Well, we give them bridging therapy, some sort of a treatment to try to keep the lid on the cancer and keep patients as well as possible. This waiting time is really an art of medicine, and we work with patients very closely. The patients are then admitted and they have a treatment phase. They get a low-dose chemotherapy and then infusion of the CAR-T cell, and the patients are hospitalized for a week. And then afterwards, they are monitored in a clinic for 1 month with a very close, almost daily monitoring. We manage complications. And then after a month, if this works, the patients have a very quick resolution of the cancer within a first month, within a first couple of weeks. The cancer starts to recede and the patients are gaining the remission. So here I just want to show you, this is maybe too complex, but let me just walk you through this. This is a timeline. So here day 0 is infusion day, and we give you a few of these cells. But if you measure them in a blood, this is a number of cells in a blood, and this is a logarithmic scale. So this is all in a lot of cells in a very short time. And why is that? Because they are alive, they grow. These cells grow in a patient's blood and become 100,000 times more than at the infusion time. But they are in a very high levels, only briefly for a few days. During that time, they do their job, killing, and they slowly go down, and then they stay in a very low levels. This is 300 days, and we still can detect these cells in some patients. This is what's called survival curves, and you may have seen this for some of your other presentations, but what basically it showed is that it is 100% of patients who are in remission, and for patients who are in a complete remission, here is in about 2 years, about 70% of these patients will stay in a complete remission. So we don't cure all patients, but we cure quite a large proportion of patients. So the response rates in, for example, mantle cell lymphoma are about 27 plus 67. So it's about 93% of the patients respond. 93%. Most of the patients have a complete response, more than 2/3. And some patients have a partial response. And here is how many of the patients will stay in remission. So 60% of patients will stay in remission. And the fact that this curve is flat means that this most likely will lead to a cure, that no longer these patients are relapsing, but they stay in a remission long term. So I did mention to you that with every new treatment, there is expectation that there are some new toxicities. And indeed, with this very unique treatment, CAR T-cell therapy, there is an increasing realization and observation of a very unusual toxicities. One of them is called cytokine release syndrome, and another one is called neurotoxicity. And they are related to a CAR T-cell expansion. So what happens in the blood of the patient that these CAR T-cells are producing all kinds of cytokines. And I think one of the good way to try to imagine what's happening in the body of the patient is to think about influenza or any viral infection, because the CAR T is inflammation. It's causing inflammation and immune activation. So one of the first manifestations is a fever, body aches, low blood pressure, sometimes low oxygen. And it also leads to a neurotoxicity, which is another side effect which can occur. And this leads to lethargy, confusion, sometimes difficulties finding words or speaking, being somnolent. And in a few patients, it can lead to seizure. One of the early manifestations of this neurotoxicity is the problems writing. It's interesting for writing how much cognitive power we actually need. So it's a very sensitive tool if you are losing that cognitive power. These toxicities occur in the first 7 days, and they are reversible. We know how to treat them. We use medications to make them go away, but they can be present in anywhere from 20 to 30% of patients, and typically they are fully reversible and completely go away. The median duration of this treatment is about 8 days, and the onset is 2 days for the CRS and 4 days for the neurological toxicity, and really explains you why we keep patients in a hospital for first week. By 14 days, typically everything resolves, and the patients are certainly often don't remember that they have any kind of a side effects. So it's important to know that because of these toxicities, there is a requirement of the patient and guardian education. And we often very, in detail review this toxicity with the patients and educate about the manifestations and what to look for. The patients are expected to stay within 30 minutes of the center for at least 1 month. They are expected not to drive for about 2 months. They need to have a wallet card with them and often a bracelet telling the caregivers, sorry, not the caregivers, but the care facilities, if they end up in emergency rooms or somewhere, they have received CAR T-cell therapy because of the different approach to their treatment. And this is an education for a caregiver who are helping us to monitor the patient once they are discharged home. So as I explained to you, all these therapies at this time are using patients' own autologous CAR T-cell. It is truly a custom-made therapy for each patient, individually, genetically manufactured. And it brings some limitations. It is incredibly expensive. As you see, you need to have a reliable family, caregivers, a means to be around our center for a month. We do provide help with accommodation, with the Hope Lodge, the free lodging, but still we realize there are significant socioeconomic and geographic barriers and access issues. our patients have to wait for manufacturing, and sometimes it's very difficult to control the disease. The efficacy is remarkable, but it's very far from being perfect. We sometimes see that the target expression is lost, and there are different mechanisms why the CAR T cell can fail. And as you see, there are significant side effects. So there is an ongoing need to improve these therapies, to modify them, and to come up with a better CAR T cell therapies. And that's also where we are with my team. And we have been very active of developing novel treatments. So in the last 10 years, the research in CAR T cell area has been incredibly fast growing. As you look here, 2017, there may be like 79 studies. Today, we have 357 studies in the worldwide, using the CAR T. They are most of the time used for the hematological malignancies. But as you see, the interest to develop CAR T cell therapy for solid tumors has been really growing, and particularly for these targets, mesothelin, which is for ovarian cancer, lung cancer, for glioblastoma, for breast cancer, for head and neck cancers, prostate cancers. These are all areas of active research and developing the CAR T cell therapy for these patients. It turned out that it's not very straightforward to bring the CAR T cell therapy from one disease to another. And particularly, there are significant barriers before the CAR T cell can be effective and successful for solid tumors. There are several reasons for this. One of the reasons is that solid tumors are slightly more complicated in how they develop, and there is this wall of the fibrous tissue, and it's somehow turned out that it's a mechanical wall as well, an immunological wall as well, and it makes it difficult for the CAR T cell to penetrate into the tumor environment. It's also other mechanisms, and there are several of them being identified, and it's very interesting to see the concepts from the basic research and how they influence the new ways how the CAR T cell discoveries are turned and emerging the new ways how the CAR T cell will have to be modified to be able to go and get into the solid tumors. So this is just a snapshot of the clinical trials of using CAR T cell therapy for other malignancies. And this is from the clinicaltrials.gov. And I can just, you can read here different CAR T cells for osteosarcoma, neuroblastoma, non-small lung cancer, B-cell lymphoma, biliary carcinomatosis, hepatocellular carcinoma, CNS cancers, pediatric tumors, liver carcinoma. And so this list is really exciting, and it looks, it is really a demonstration, and it is exciting to see the rapid development of the CAR T cell therapy across the oncology in general. At this time, none of these treatments are approved by the FDA, and they are only being used in the clinical trials. So at the University of Minnesota, we have many clinical trials. I am most familiar with the clinical trials for hematologic malignancies, and there are several concepts being developed here. One of the most, I think, promising and exciting is using the off-the-shelf allogeneic immune effector cells. This is a healthy CAR T cells or CAR NK cells, which are from the healthy donors, being collected, harvested, frozen, and ready to go for patients who need. This eliminates that waiting period of time. It also eliminates the need for manufacturing of the patient's own T cells and allow us quicker and more efficient treatments. We have NK cells therapy for leukemia, lymphoma, myeloma, NK cell CAR for AML, CLL. We have a CAR T cell for prostate cancer. There are some immune engagers for ovarian cancer, breast cancer, and lung cancer. And there are some new treatments which are being developed for colon cancer. But I do not have a detail or where the clinical trials are for the solid tumor malignancies. But if you are interested and there is certainly an opportunity to be referred to solid tumor oncology with expertise in cell therapy, to have a discussion and to see whether any of our clinical trials would be applicable to to you or the patients that the patients currently need it. So I wanted to make sure we have enough time for questions. The field is complicated, and I hope that I have shared with you some of the new concepts. The CAR T cell therapy right now is for our patients without any age limit. And this is important for stem cell transplant. There is an age limit for some of the chemotherapies. There is an age limit. We feel that CAR T cell therapy can be for everyone. And it just depends on the consultation with the team to see what is the best fit. For all the patients we see, we do a frailty assessment to assess their physical strength and their organ function. We look at the prior comorbidities, at prior therapies, at your fitness. We look at the tumor bulk, of course, the assessment of the cancer and the expression of the target. And then we have teams which comprise physicians, nurses, social workers, physician assistants, dietitians, physical therapists, occupational therapists, and together, and the research nurses and research person, and together we come up with an optimal management and often recommend investigational cell therapies if we think that there is a successful path and hopeful path forward. So with this, I would like to just mention physicians who are active in this area at University of Minnesota, and that these are the experts in hematologic malignancies. We have a number of other physicians who are experts in solid tumor oncology, and all of our entire team is dedicated to improving the treatments options for our patients. So with that, I am happy to take questions or entertain any comments you may have. And I really appreciate that you have joined me and that opportunity to review this complex topic. But I think that you will hear about this topic in general public newspapers as this has become one of the terms which is gonna be used in oncology in general. So thanks so much again.
Thank you so much, Dr. Bachanova. That was such an insightful presentation. I learned so much personally, and I'm sure folks in our audience here have learned some things too. It was really exciting to see some of just the progress, the recent progress that we've seen is just leaps and bounds. So that really stuck out to me. If anybody does have questions, feel free to start submitting those into the chat box, and we'll get to them in just a quick second. Before we move on to the questions, I just wanted to give everyone the reminder that a survey will be emailed to you tonight. So don't forget to do that. You've got the chance to win a swag bag of Cancer Center goodies. So please definitely keep an eye out for that email. And it looks like we do have a few questions starting to come into the chat box here. So the first question, it looks like Dr. Bachanova, that came in is this a therapy that could be used instead of surgery?
Yeah, probably not. The surgery for many cancers is still often the most effective way to get rid of the area which is the most bulky or problematic. So not yet. And I don't think it's going to replace surgery, but it may be used to clean the margins around the surgical bed once you remove the bulk of a tumor. For the diseases I mentioned, the surgery is not an option because they are typically whole body diseases. But for some cancers where the surgery can remove a tumor almost full of it, the surgery still is going to be very helpful. But yeah, I can imagine just in sort of a theory that CAR T-cell therapy can be used as a what we call adjuvant to clean the bed and to get rid of the cells which may be hiding, and you may not be aware where they are hiding or how to get rid of them. Good question.
And along those lines, Dr. Bachanova, can you talk a little bit about how folks might find out if cellular therapy is the right option for them?
Yeah, I would start with your oncologist, and depends on your particular situation. And if oncologists feel that you may benefit from the consultation, it really requires a consult at the University of Minnesota. So to be referred to one of the specialists at the University of Minnesota, the cell therapy BMT program has those specialists, but I would start with a local oncologist to talk to them and maybe request a consultation or second opinion at the University of Minnesota or Mayo Clinic. These are the two places which probably are having these clinical trials available.
And what does that process look like to be considered for a clinical trial?
Yeah, thanks for that question. It starts with a conversation with the expert in your disease area, with oncologist. And clinical trials have a certain criteria for the eligibility. So that requires to knowing, what prior therapies the patient had, whether they worked or not, what is current disease state, is the patient in remission or is the disease coming back? And then one of the important aspects of the clinical trial is that often, just because there are three, let me just kind of explain this. There are three tiers of clinical trial. The first tier is called phase 1. And phase 1 is when we use the agents the first time in humans. It's called really first in man sometimes. And the purpose of these clinical trials is to understand what is the dose which should be used. Sometimes there are different levels for dose escalation. What's the toxicity profile? And it always has the chance to work. I mean, that's the hope, and that's why we are using it in a phase in patients. But there is no guarantee it's gonna work. And that's what phase 1 is asking: is this therapy effective? Everything which is moved to phase 1 has a preliminary success in animal models. It's cure animals. Often it cures primates. It's effective in animal models. That's why it's ethically say, as ethical to use it in humans. So phase 1. Phase 2, we know it's safe. We know the dose. And in phase 2, we know in a phase 1, it's actually worked for some patients. It's preliminary effective. In phase 2, it's usually a larger study. And in phase 2, we are learning of how effective it is for this particular disease. And is there a disease subset in which it maybe works better? Phase 2 is often where we expect this treatment to actually deliver clinical benefit, significant benefit. So all the studies I showed you with the CAR-T and the studies I had for CAR-T were phase 1 and phase 2. For phase 3, those are the large studies involving hundreds of patients. And typically they're asking this question: is this treatment, which was found to be very effective, the same or better than currently approved standard of care? So let's say there is a chemotherapy which is used for breast cancer because it works in half the patients. That's what we have. That's the best we have. So we are using it on our patients. But we can have a treatment which seems to be better, or we think it's better. We have to really make a randomized study to compare it to this standard to find out whether it's better or not. And some phase 3 studies are successful, and this new therapy then becomes your new standard of care. That's how we make progress. So clinical research is complicated, but this is the only way how we can make progress, and the only way how the confidence can be reached in using the treatments in a larger and larger number of patients. The number that the patients I have treated, often they come to us because they have no good options, and they are willing to take any options with the hope that that can work. And that's what I was talking about, the courage and commitment to realize that the treatments we use now are here because of the courage and commitment of the patients which we never met before, but they were here for the future patients who can benefit from the treatments as well. So this is the complexity of the medicine, and we as oncologists are really always looking forward and not willing to give up for the opportunity to help patients with a new therapy.
Thank you so much for that. That was a lovely answer. And you talked a little bit towards the end of your presentation about side effects. I'm wondering if you can talk a little bit about what we may know about late effects of CAR T cell side effects.
Yes, the late effects of CAR T is an area of active research. For what we know now, most patients recover quite well and do not experience late side effects. And even the infections often are happening in the first couple of months or a few months. The late side effects are what we are thinking maybe months, years, or decade after treatments. And for right now, there is really not yet any signal of worry that there will be a long-term side effects. One of the issues is that the patients already have lots of treatments. So if there is a risk of secondary cancer or secondary malignancy or some secondary leukemia, it's difficult to say what is the culprit because patients often have so much therapies before. But it certainly is one of the concerns, and that's the reason why the monitoring of CAR T-cell patients is recommended for 15 years. The other things is late infections, and there are some patients who may experience late infections, and we do watch for those and are on a monitoring for at least for a year.
Thank you. I do want to note I'm getting a lot of messages, both in the public chat and directly to me. So I apologize if we are not able to get to your question today, but I'm getting through just as many as I possibly can. There is a lot of tremendous feedback on your presentation, Dr. Bachanova. Lots of people have learned a lot and felt it was easy to understand and are really just singing your praises. So thank you again for all of your great information.
Thank you. It means a lot. Thank you for all the nice words. I appreciate it.
Yes, thank you, thank you. Keeping an eye on the time here, I'll ask you maybe one, maybe two more questions. One question here that's come up a couple of times is, do you see the future including other autoimmune disorders such as rheumatoid arthritis or other disease types?
Yeah, I'm so glad whoever asked this question. Thank you so much. I totally omit it because we talk about the cancer, but this is so important. The CAR T-cell therapy does have a completely new door opening for the patients with rheumatological disorders and autoimmune disorders. And there is number of new clinical trials. There are a number of new clinical trials which are opening at the University of Minnesota and others for lupus nephritis, lupus erythematosus, rheumatoid arthritis, SLE, and others. And the premise here that CAR T-cell can reset the immune system in a way that autoimmune antibodies are eliminated from the blood. And in fact, for the first time in the history of rheumatology, the patients may be taken off their immunosuppressive medications, and in fact can hope for a long-term remissions of their rheumatological malignancies. There was one of the first reports of observing this was in a science journal or Nature. As you know, this is one of the top-ranked journals in medicine. And there was case reports of, I think, a handful, maybe half a dozen of patients who got in a long-term remission with the lupus. So very important indication, future indication potentially for the CAR T-cell therapy.
That is super, super exciting. I think that we are all very eager to learn more about cellular therapies and the impact that they're going to have. So just our last question here before we wrap things up, what can we do to better advocate for more research and better access to cellular therapies?
Yeah, it's a good question. It's a good question. I, as a patient or as advocates.
I think both. Yeah.
I think it's all about education and understanding that we are only here together. We have to have a really ownership of our faiths in every aspect of life, and being understanding that we are here together, both scientists, clinicians, nurses, researchers, patients. We are here together, and I think educating all the layers of our wonderful team is the best way how we can bring awareness, bring that each of us can contribute at some level. And it all means a lot for patients who are at need. So I think your series is a wonderful example of where is all meeting with a fruitful results and a success of delivering that education publicly. And I really appreciate the opportunity to be here tonight.
Amazing. Well, thank you again so much. We are out of time here for tonight, but as a reminder, the recording will be available on our website by the end of this week, and you'll be able to then review it again and share it with others if you think that you'd like to get them on the bandwagon as well, so we can help build that community around cellular therapies and cancer research and treatment. As another reminder, you will receive that email tonight with a quick survey that really helps us get your feedback and continuously improve our programs. You also have that opportunity to win the swag bag of new stuff from the Masonic Cancer Center. And then next week is the last session of our 2023 Thrive series. So we hope that you'll be able to join us next week, Tuesday, November 14th. In recognition of National Family Caregiver Month, we're proud to host a session all about caregivers, cancer caregivers, and their role in cancer survivorship with Allison Brenniger and Pamela Mason. So we hope to see you then. Thank you so much, everyone. Have a wonderful night.